I'll bet there aren't very many of you out there that do have a degree in botany. That's kind of old school at this point. Can you hear me at the back of the room? Oh, good. Okay, so what I decided I wanted to focus on today was my students and their work, because the invitation was from students. There is no higher honor when you are a researcher than to be asked to give a seminar for students by a student organization. And I think that the reason for that is largely because you're all trapped in classrooms listening to terrible presentations most of the time. So if you like somebody's presentation and you want to hear it, it's just a huge honor. So I really appreciate the invitation. So you can see on this front page that what I'm going to be talking about is data-driven perspectives in the plant sciences, and really how to enable you to use those perspectives. The lab group that I work with is at the bottom, and as I go through some examples, I'll try to call out which students and other lab members have worked on which projects. Before I get started, I want to do a little bit of an advertisement. We have a P3 program at Iowa State now that many of you know about. It's a graduate program across 5 different existing majors. And the idea is that we pull together students that are interested in data science, plant science, and engineering, and give them an education that uses a little bit of each. And so it is a really sort of broad topic. And for any of you that are interested to know more about that, there's a poster upstairs. A number of the P3 current group cohort is here, and we'd love to talk with you about it. So the outline of my talk, first I'll tell you what my group works on and how we pick projects, and then I'm just going to go through some examples that hopefully I put them in an order so that it seems like a story. And then at the end, if I can get there fast enough, I want to talk with you a little bit about how to frame and advertise science, both to people that you want to give seminars to because they're scientists as well, and to the general public and what the differences are. So why share data? We think that information has to be communicated or it's effectively lost, and the results should be reproducible. That's kind of a thing that we say that's what science is, right? It's reproducibility. You get this every time. That's true in the lab, it's true in the field. It turns out it needs to be true on the computer as well, that when somebody does an experiment, it needs to be something that you can demonstrate to yourself that it worked for you too. So there was this editorial that came out that was a fairly famous thing among people like me a little over a year ago on data sharing. This is in the New England Journal of Medicine. The excerpt here says, someone not involved in the generation and collection of the data may not understand the choices made in defining the parameters. That's true if you didn't write your materials and methods well, right? It would be hard to figure out why somebody did what they did if they didn't write it up well. At least that's how I would interpret that. So it turns out the authors, if you read through this paragraph, I'll give you a moment, focus on the blue part. Some of you may not be able to see it from where you're sitting. I'll read the blue part. There is concern among some frontline researchers that the system, and the system here is being able to document and give out your data, the system will be taken over by what some researchers have characterized as research parasites. And so my hope today is to tell you there's lots of data out there. You can use it, and when people document it well, you too can be a research parasite, and I'm going to go through and give you some examples of how to do it. So in my own group, the way that we pick projects is we look for things that are helpful to other people. And one of the ways that I try to explain that is through puzzles. And scientists love solving puzzles. There are lots of different ways to do it. Some of you probably do this at family holidays. So you start out with a puzzle box, you flip over all the pieces, and then there are people that focus on parts of the picture. There are the people that go and make the outline. And then at the end, hopefully it all comes together, right? So if you think about the different scientists you know, there are people that do big grow outs where you go and you look for a mutant. Maybe these are the people that are turning over all the pieces. There are people that are interested in some peculiar aspect of biology that when you listen to their seminars, you think, how did they end up getting interested in that, right? And what it is that my group works on is that frame, and that is being able to give people something so that they can take their biology, their interests, their science, and put it together into this lovely picture at the end. The problem with science, of course, is that we don't have a box to look at to know where we're going. And at the end, we find out that the dog ate part of the pieces, and we're never going to get the whole picture. So in my group, by building information platforms and developing data sets that are available, they're large-scale data sets, it helps the whole endeavor. We provide a framework for data sharing and help others to be able to put their chunks into that big picture. So how does that square with crop improvement? So crop improvement is dependent on phenotype. We select the phenotypes. In my group, we usually start with genome sequences. So the sequence allows us to find genes, the sequences of genes. We have a genomic sequence. We do gene calling. And then from gene calling, we can start to predict what functions are. And then those things get expressed in the environment. You get to a phenotype. So what I'm hoping to do today is to take a number of different examples in my group to show you where they fit into this overall architecture of how we get to crop improvement, starting with genome sequence. The examples that you're going to get are from the past, some work that I did when I was with USDA ARS, working at Maize GDB. There was a tool we developed called Locus Lookup, and then we're going to progress through to talking to about a current large-scale project called Genomes Fields. So let's start with maize reference genome status. Everything I'll talk about today is maize. It could be any species really that you would do these kinds of things with, but I'll focus on maize. So the first version of the whole maize genome, RefGen V1, was produced in 2009. And that sequence came from cloned backs. So these are chunks of the DNA that got sequenced and assembled back together. In 2010, computational improvement to that, a better assembly of those same sequences came out called RefGen V2. And then in 2013, RefGen V3 came out. And that one, they did add some more sequences still. The main backbone was from cloned sequences. And the thing you need to know about these cloned sequences, that little bits and pieces of them, the order and orientation of them is not actually known. So if you look at a pseudomolecule sequence, that's the sequence you have for a chromosome from one end to the other, there are lots of pieces inside of that, but we don't really know if that's the order and orientation of those pieces, and that's really hard to deal with. B73 RefGen V4 is available now. I have it grayed out on this slide primarily because it's not stable yet. So if you want to discover something new, it may be useful to you, but if you're really trying to work in a genomic context and you want to have a stable genome to work with, in maize we're still using V3. A place to get started looking at a genome sequence would be the model organism database, if the species you work on has one. Maize GDB is the model organism database for maize. And I could give a whole seminar on maize GDB and never get to the end of it. I'm not going to do that today. I'm going to focus in on one tool. That tool is the Locus Lookup tool. When you have a genome, you want to know where's my gene, right? So if you start with a genetic map, you have your gene here. I call it unknown 1. For maize, because we have these cloned sequences and we have a physical map, we might know for my gene what some markers are that are on the left and the right of it. And there's my genomic region. Well, it turns out those cloned backs that are our assembly in maize, they are, in fact, distinct fragments. And because these fragments are in who knows what order and orientation most of the time, my unknown gene, even though I would like to think that it's up here in that middle back, it's possible that middle back doesn't have it. It's possible that, it's possible that it's out here on the end of this back, or it could be out here on the end of this back. And so if you're looking for your gene, you would like a computational tool that delivers back to you that entire region so you don't miss it. So we developed a tool that does just that because we talked with a number of different researchers that were doing this work, and it would take them a day to go and assemble all of the information together to figure out, you know, is it this back? Is it these pieces? And so Locus Lookup does this for you. The one that I'm showing, the gene that I'm using here to give you a demonstration is GA1. And if you are interested in its function, I would refer you to a poster upstairs that's Ryan Huffman and Paul Scott. I am happy to tell you that when I got here today and I said, I was hoping this was an uncloned gene. Is this still an uncloned gene? And they nod their heads, yes, we still don't know what it is. So it's still a good example. So for Locus Lookup, I want to know what is the genomic region that might contain this gene, GA1. What gets returned to me is an answer that says There's not a gene model associated with GA1. It's not been physically mapped. It's not in a place back that we know of. It has been genetically mapped, and here are some markers on either end. And then I have this at the end that tells me exactly where it's placed. You can do this not just with a single locus, you can also do that with a couple of markers if you have a QTL, for example. So you can run locus lookup on both of those markers and come up with a region. So let's imagine that I have these two markers, and I know there's a region of the genome, and it's for some trait I care about. Here is my locus lookup, my locus pair lookup result. And at the bottom here, what you see is the genome browser, and what's shown in green down here is gene models. So let's zoom into the genome browser. There's our track with gene models. If we zoom all the way in, then it starts to look like what we think a gene model looks like. So imagine how many genes are actually in that region that's somehow associated with my trait of interest, plant height. It's a lot, right? So a question is, once you come to a region like that, if you wanted to know what gene causes my phenotype, how do you start to pick that out? There are lots of different ways to do it. But I'm going to show you one that we've been working at creating a data set to help you narrow down to a gene that may be of your interest. So next, I'm going to talk with you a little bit about functional annotation. This is the work of Gforces, who is in my lab. How do we assign gene function? So if you do a computational prediction, the pro is that it's really fast. The con is that it didn't really tell you if that's what that gene really does, right? If you work in a wet lab and you want to find out what this gene does, the pro is that you're going to end up definitively figuring out, hopefully, what it is that it does. But it's very, very slow. So we do computational functional annotation as a prediction so that people can work from there. The way you do that is to start with some experimentally characterized gene that already has high confidence functional annotations associated with it. Then you do a sequence match so that uncharacterized genes can get matched to their experimentally characterized counterpart. Then you can inherit those predicted functional annotations. Most of the time when people are working on this, they use the gene ontology. Go. What is the gene ontology? It's a controlled vocabulary, and I warn you in advance, I'm well aware that most people, when I start launching into ontologies, they think I'm going to die in this room. Don't worry, it's not going to take very long, and the upshot is that we have something for you that may be useful. So a gene ontology is a controlled vocabulary. The terms are hierarchically related. I'm going to show you a little bit about how they work, just so you understand what the results mean. So here is this graph, and you see a term at the bottom, cytokinesis after meiosis. The way the graph works is at the top of the graph is the most general term, at the bottom of the graph is the most specific. This specific term at the bottom is called a leaf term, and leaf terms, if you assign nothing but that bottom term, everything upstream gets inherited. So that's one of the useful parts of a gene ontology. The Go comprises three different graphs: molecular function, biological process, and cellular component. So see, these are three different aspects of gene function. One thing that many people are unaware of is that functional annotations have bits of information attached to them that allow you to know how the assertion that that's what we think this gene does was made. So some of the information will tell you this assertion is made with experimental evidence. That's golden. You want that. Sometimes you get something that says automatically assigned. That means it got cranked through some pipeline and nobody looked at it, and it's not even clear what the original data set was that we inherited all these terms from was. So that's like the worst case scenario, right? If we look at these terms in Arabidopsis, we see some very nice, directly functionally characterized term associations. We know what genes do in Arabidopsis, or at least some of them. It turns out in maize, the data set that most people use, which is from Gramine, is almost entirely automatically assigned. And so that should tell you something about your confidence in using the Go annotations right now for maize. So what we did was to put together a pipeline called Gamer so that we can improve those annotations. We want better functional hypotheses. So the way we did that was to only use high confidence evidence codes for input. We used a lot of different methods to predict gene function, and then we evaluated the output using a gold standard, and we're enabling the community to review so they can say this is true, this is false. So what we wanted to get out of it is this data set so that you could use it. So the data sets, now that we have a data set called the Gamer annotation. It came through a pipeline that I can talk about any of you later if you're interested. We want to compare it to Gramine, which is the one that many people here probably have used, as well as phytosome annotations. And we're doing that based on, like I said, a gold standard. The gold standard came from MazeGDB, and these are term assignments that were created by curators based on experimental evidence. The number of genes in that gold standard is about 1,600, and there are more than 2,000 annotations. The most annotated one in the gold standard that I'll use as my example is Nanoplant1, which has a dwarf phenotype. There are 7 leaf Go terms associated with it. 2 of them are pretty obviously associated to plant height. The first one is brassinosteroid biosynthetic process. The second is unidirectional cell growth. You can imagine how those 2 things contribute to the height of a plant. You're going to see some graphs in the next couple of slides. Let me show you how to read them. So this graph that you see here is the data set. The nodes are labeled in D, and it's blue. Our gold standard, they're a different color, and it's labeled with a G. Leaf terms have that leaf next to them. And then we have an overlap. If a prediction is in both the data set and the gold standard, then it turns into this green color and shows you that that's a match. Then we want to evaluate, so we evaluate based on F-scores. A high F-score is mostly green, so this is good. It means that the prediction is very close to what the gold standard says, and a low score looks like this. A lot of these gold standard terms are not matched in your data set, and an intermediate is a mixed graph, like you see in the middle. So if we look at Nanoplant1, what does it look like for phytosome? That data set has nothing. What does it look like for Gramine? You can see that there is some overlap. There's a lot of what the gold standard annotation looks like that is not found. If we look at Gamer, there's a lot of green. It is a much better, for this individual gene, it's a much better annotation. Okay, so I just showed you an example. What does this look like genome-wide? I forgot this, sorry. So if you look at those leaf terms that I mentioned to you, brassinosteroid biosynthetic process, that term is right here in the Gamer graph. It's right here in the Gramine graph. They both did pretty good there. If you look at, instead, unidirectional cell growth, the Gamer data set got that one, but Gramine did not. So over the whole genome, what you see, for the percent of genes that got some kind of a Go annotation, if blue is showing you Gramine and green is phytosome and red is Gamer, under cellular component, we have a whole lot more genes covered. The same is true for molecular function and biological process. This is a giant improvement on that, in that regard. If we look at the number of annotations, so you can have one gene, it can have one or more annotations. The number of annotations is also higher for our Gamer data set. Okay, what about the quality? Is it any good? So if you calculate F-scores for those overlaps, this is Gramine, phytosome, and Gamer. Gamer is either better or very comparable in quality to what it is that you see in these other existing data sets. So this is something that's available now, that's much higher coverage, and it's similar quality. So let's go back to the reason we did this, right? Which genes in this region are important for plant height? Wouldn't it be helpful if you had some annotation to some one of those genes that said unidirectional cell growth and brassinosteroid biosynthetic process? You would bring that to be the first thing that you would want to see, is that my gene of interest. So that's the purpose of this. Next, we're going to talk a little bit about a CRISPR-Cas9 prediction tool. I would ask, what do you do when you get to the same point in your process if your gene had not been functionally characterized, or even its functional characterization predicted? If you have some gene you want to work on, you might try reverse genetics, knock out gene function, see what it does. If you have a functional gene, you have an unknown phenotype, what is it doing? If you have a mutated gene, what's the phenotype? And it's going to tell you, this might be what the wild type function is. You maybe can interpret it. So in my group, we have a tool that we've been putting together that enables you to predict CRISPR-Cas9 sites. I am not going to go into the detail of how that works because I think that's going to be in our next seminar. But in effect, it is a way to create a mutation in a very specific way. The tool that we put together is called Cgap, and with Cgap, which is available at this URL, you go through steps to give it input, and the input is what do you want to cause a mutation in, and do you want, what size do you want to make your guide RNAs. Then the next thing that you do after you've picked your sequence, you give it a size, you deal with some other parameters, you analyze it. Not only do you get the analysis of your region of interest, what would be a good guide RNA, you also, the whole rest of the genome gets looked at if it's a genome that's been sequenced, so you can tell whether there's any high possibility that you're going to end up with off-target problems. And so the results that come out down here tell you similar sequences in the rest of the genome. That has been published, and you can find that online. So let's say you have a gene, you want to do that to it. What genome are you, what germplasm are you going to break your gene in? And I would tell you that probably you want to do it in B104 or something else like it. The reason is because our line that has been sequenced, that is our reference genome in maize, B73, is one that can't be transformed. So that's a problem, right? All of your tools are in B73, you can't transform it. So the question is there a line that's very, very similar to B73, and yet it can be transformed? The answer is yes, it's B104. In fact, they're both derived from the Iowa stiff stock synthetic lines, and B104 is very nearly identical at the level of nucleotides. If you look just at SNP regions of the genome, the SNPs are 93% identical, and those are expected to be variable. So they are very similar. So we did sequencing with Illumina pair-end sequencing to 50x coverage. We did some assembly, both reference guided and de novo, compared it with B73 RefGen V3, called gene structures, and also called annotations for functional annotations. And those are available now at MazeGDB. This is work that was done by Nancy Manchanda in my group. So if you go to MazeGDB, you can find B104 sequence now. We've done these structural annotations. You can see the Go predictions, and we've also, using the Cgap tool that I showed you, predicted CRISPR sites across that entire genome. We do know that we would like to have a better assembly. We would like to have a better sequence, and so right now we're doing PacBio sequencing, and then we would assemble, annotate, and also release again at MazeGDB, and that should be available by summer. The next project I want to tell you about is one that Ming in my group has worked on, and it's G quadruplexes. This is going to be a lot more like if you have a research lab, what you would do with all of this information that's out there. How would you use it to solve your question? So G4s, this is Ming in the picture, G4s are a DNA motif pattern. From the 5' end, you see 3 Gs and then a loop of something, another 3 Gs, a loop, another 3 Gs and a loop, and you build it like that. So those Gs bond to each other, and they're stabilized by a cation, and that happens throughout the structure, and there's a bulge out on the other strand of DNA. So these generally are structures in the DNA that are single-stranded structures. What do they do? So a group of us did some work trying to figure out what they might do, and what you see in this upper right-hand corner is mainly to remind me to tell you that we mapped G4 containing genes to their various different pathways and figured out that they are highly associated with stress pathways. So what do we think they might do? We think that if they're in a promoter, they might block transcription. So if you think of where that G4 is shown in this image, you have the template strand shown in red. Promoters are here, and they could get in the way of RNA polymerase. They also, if they're on the other strand, here's the template strand, here's the non-template. Once RNA polymerase is actually hooked on, they could get in the way of RNA polymerase and keep those genes from being transcribed. You can imagine inside the cell, if you have stress genes, genes that you want to induce with stress, it would be great if you had all that transcriptional machinery poised and ready to go once you hit stress. You resolve that G4 and ta-da, suddenly you've got transcript, right? So the other place that we think that they could be functional is if that G4 is actually after the start codon, then they would also likely occur in the mRNA. If they're in the mRNA, it is possible that just like I explained for transcription, for translation, you have the transcript sitting around in your cell, and maybe the translational machinery is ready to go. We don't know whether any of this is true. This is just our model for what we think might be happening. So Ming and my group has started looking at RNA-seq data, and what he did was to look at the frequency of genes with no G4, which is shown here in blue, and the frequency of genes that have G4s, shown here in red, and what their expression is under submergence conditions. The x-axis on what you should see here is showing you fold change differences. So fold change can be increased or decreased under stress. So anything that's at 0, it's basically the same expression both during stress and non-stress. When you start to see something that spreads out in that graph, and it's on the left and it's on the right, it's responsive to that stress in some way. So if we look at submergence, what we see is that these G4 genes are more responsive to stress, positively and negatively. What happens with other stresses? Well, it's basically the same pattern. You see it with heat, salt, cold, and UV. So it turns out that 42% of G4 genes are differentially expressed versus only 26% of the non-G4 genes. What does that mean? We started looking at co-expression graphs to try to figure out what does that mean, which genes are these and what are they doing. So if you start with me on the upper left here, you see in rows genes. You see in columns of this matrix different samples or different stresses. And for any given gene, here we have gene 1. Here's this sample, this sample, this sample, and we have expression differences. Oops, sorry. Then when you start to look across different conditions at how that gene is being expressed, you can start to see which genes their changes are similar. So if then I make a matrix to the right, and you see those same genes across both the X and Y axis, you can see that one gene versus itself all along the diagonal is 1, and then things that are highly correlated have higher numbers, and low correlations for their co-expression have low numbers. You come up with a significance threshold, and then you turn those into 1s and 0s. You're going to say this one is correlated, this is not correlated. And then from this graph of 1s and 0s, then you can start to draw a network where each gene is a node. And if the two genes are co-expressed, they get an edge between them. Why does this matter? This matters because those G4 genes, as it turns out, are party hubs in stress networks. So if we look at a non-stressed tissue, and this is our network, we look at a stressed tissue, here's our network, and we subtract out those networks that are non-stressed tissues, we end up with a stress-induced network. And that red dot here is the one that has a lot of different lines drawn to it, and that's what we call a party hub. And very often these are genes that interact with lots of other genes to be able to regulate gene expression. So it turned out that these party hubs have a G4. It happens to be mainly on template strand. These are party hubs, and many of them, as you might expect, are transcription factors. So if we looked at just G4-associated transcription factor activation, it turned out that if we looked at these different stress types with non-G4 and G4% genes responding to that stress, it is a higher ratio for G4 over non-G4, telling us that these G4 genes are in fact doing something different. And this is specifically the subset that is transcription factors that have G4s on the template strand. So if we go back to our model, here we have the RNA polymerase in the middle one. We blank this out. This is what it is that we think we have seen. We have seen something that tells us that this part of our model may actually be occurring, and that is that perhaps the G4 that is in this location is actually functionally relevant to this response to stress. And perhaps it's through this model that we've developed that it's poising the transcriptional machinery ready to respond to stress. So hopefully by this point in the talk, all the stuff that you can do with analysis of other people's data, you think, wouldn't it be great to have a big data set that was genotypes and phenotypes and environments, and you could do all this stuff on your own, right? If you're sitting there thinking that, then you might be one of those research parasites. So it turns out that data set is available, and it is a data set that's been put together by an organization, a group of us called Genomes to Fields. Let me tell you a little bit about that. So Genomes to Fields is a project that lots of people are on, and as I progress through this section, you're going to see lots and lots of names. This is the group of people on the screen who've been specifically involved in data management. The objective of Genomes to Fields is to take genomic information, phenotypic data, and environmental data and allow other people access to it so that we can develop a better working knowledge of how maize performs under lots of different growing conditions. There's a G/E subgroup that basically is developing these methods and resources so that we can predict phenotypes for diverse environments. These are environments we've already seen, environments we expect in the future. The kinds of things that we've been doing are to assess environmental effects on the germplasm we've been growing in lots of different places. So 22 environments and 17 states in Canada have been evaluated. Here's a map showing you where collaborators have been growing fields. We have genotypic data. We have environment data, including weather stations, GIS data, soils. And we have phenotype data, both core traits and that are listed here that are aspects of plant morphology, ear morphology, agronomic traits, and productivity. We also have even aerial images to be made available. We've been managing data on Cyverse and releasing data through Cyverse. We've used the Cyverse discovery environment for dealing with allowing people access to the data as we go along. We've used a wiki to put all of our methods, and in some ways, I should say, we've also used Ramona Walls from Cyverse, so she's contributed a lot of her time to helping us be able to make the Genomes to Fields data available through Cyverse. So for managing the data, the things that we've been focused on are developing ways to streamline data collection and analysis and access. That can be something as simple as we gave everybody the same methods to go out and measure plant height. And it turns out that was not straightforward. We had lots of protocols. We settled on protocols that had been developed at CIMMYT, and then we sent these protocols out to people and realized that even with those protocols, they were going to measure all these plant traits quite differently. So one of the first things we did was to add a lot more detail to all of our methods, and everyone did it quite well. We've also been in the process of what I would call building the ship as we sail it. So there's not a really good method for doing all of these kinds of things. Any of you that have ever tried to manage field trials probably can think back very easily to all of the emails with attachments and spreadsheets and trying to get the information where it goes, and it can turn into a real nightmare. The last guiding principle is that we really are focused on releasing data annually. So this is our first release that I'm showing. There's a DOI for the 2014 data. If you just search for Genomes to Fields online, you're going to find that. As of today, we've got the DOI for the 2015 data, and we have an internal release ready for 2016 for collaborators. So the way that we're releasing data, we have the field season shown in this first column on the left. We have a release to collaborators that occurs within the following year, and a public release of all data via a digital object identifier the following year. What that allows is for us to be able to find issues in the data sets during that year that we're still looking at it internally, and it also allows us to get a little bit of a leg up on doing some of the analyses ourselves as a group. So there are lots of G/E cooperators at a lot of different places. The Genomes to Fields collaborators is an even larger group of people. That project is led by Natalia De Leon and Pat Schnabel. And the sponsors for the Genomes to Fields initiative are varied and sundry groups, right? It's taken an awful lot of effort on the parts of some people that are involved in this just to coordinate, making sure that everybody is supported. So I'm getting toward the end here. How does building data sets and information platforms relate to crop improvement? I've tried to map onto here the little bits that I've told you about where I think they belong in this continuum. And it's all along. So there are parts of this that relate to different levels of data management. Do you need a tool or do you need a data set? The answer is you need all of those things. And to be able to get to the point of crop improvement, we really do have to have projects all along that continuum that allow you to be able to interact with the data. So the last thing that I want to tell you about today is something that I have only recently been paying much attention to, and that's how you communicate science. And so it's basically framing and advertising, and I would say in a cute way, eat your GMOs and believe in climate change. So if you want to get impact, if you want to frame and advertise what it is that you've done, there are two different groups usually that you'll be talking to. There are others, but two basic groups. For our colleagues, we tell each other what we did, and we're fascinated, right? You have the data for something, I'm going to believe you if you show me the data. You publish it, you attend meetings, and then you start delivering seminars, give posters, tell everybody what you did so they can make use of your work. So the public, it's not the same. So you have to think about where they're coming from and try to see from their perspective if you want to communicate, and tell them why your work matters to them, not you. And that's hard. So what that takes is respectful engagement, and this thing I mentioned, framing. So here's the first example. There is a sentiment that is an anti-GMO sentiment. There is psychology behind it. People understand why people are anti-GMO, and it's called essentialism. What is essentialism? Here's an example. People think of the DNA as the essence of an organism. This is the core of where it comes from. It's its genetic code. If I'm going to put a fish gene into a tomato, I have breached species boundaries. This is problematic conceptually if you're not a scientist. This probably is teleological and intentional thinking, and it assumes that nature is a beneficial agent. So if you think of all of the animals that are out there to eat you, and all of the diseases that you would like not to have that are all perfectly natural, you know that nature is not, innately beneficial. But in fact, people do think this. So then you will start to hear people talk about how making GMOs is somehow unnatural. Perhaps the scientists are playing God. Another aspect of this anti-GMO sentiment is disgust. It also is based in teleological and intentional thinking. And you see things that say GMOs can cause disease and sterility. They can contaminate fields, food, and feed. We know that this is not the case, but this is what you see. And so the public sees all of this information that's out there, and their perception is that GMOs are dangerous, potentially even immoral. So if you think further about how it is that as scientists we very often talk about GMOs, we talk about herbicide resistance and pathogen resistance and increased yields and increased shelf life, and nobody cares. You tell this to somebody that is unhappy about GMOs, and this didn't speak to them. So it turns out when you have engineered crops, you think about these traits, it's hard to explain to somebody why they should care about those traits. For the public, we need to highlight examples that directly benefit the consumer. Some examples are no cyanide cassava, reduced lysoalanine soybean, decaffeinated coffee, and reduced allergenicity peanut. I personally have told stories about reduced allergenicity peanut because I have a niece that is allergic to peanuts. And it turns out that talking to people about things like that, it really does make them think this can be used for good. So let's talk about climate change now. How do we frame climate change where somebody pays attention that's in the public? The way that we scientists do it is we say we're all going to die, right? You've seen this in every proposal you've reviewed lately. You've seen this in every paper that you've read lately about how we have to do this better than what we're doing, or by 2050, we're all going to die. So that is not the way to talk to the public about climate change, it turns out. And where do we find that out? Just like the GMO argument, it comes from psychology and sociology. They're our partners in science out there trying to figure out how to get through to people on data. So we need more food. We know that we need more food by 2050. But it turns out that if you're trying to get through to somebody that is a conservative, that the conservative mindset generally is they want to keep things the way they used to be. And if instead of telling somebody what's going to happen 50 years from now, you say to them, do you remember back when all of the maps showed you that you could plant your tulips in February and now you can plant them in January? What happened? Wouldn't you like to be able to do things the same way that your parents did? Wouldn't you like to be able to do things the way your grandparents did? They will say yes. And if you show them that in fact what it is you're trying to work at is to get back to that same place where we used to be, that seems to make a better impression than trying to tell people we're going to die 50 years from now. This has been also reviewed by the LA Times, not just in the PNAS. And they say to mobilize Trump's America for environmental protection, invoke the past. So the next time you're talking to somebody about climate change, maybe you don't want to use the word climate change. Maybe you want to talk with them about how the weather has become more variable and how planting dates have changed and that you would like to go back to the way things used to be. So to recap, I told you a little bit about what my group works on, how we pick projects, some examples in my lab, and started back with Mays GDB and worked all the way down through GMs to fields. And we looked at framing and advertising. In my group, like Genomes to Fields, the funding that covers our group is various and sundry. The key collaborators for those projects I talked about in this talk at Iowa State University, Ido Friedberg is someone that's worked with us on gene ontologies. Can Wong has worked with us on the B104 and CRISPR efforts. Sean Dorius from sociology is my partner in crime for trying to figure out how to communicate science. At Mays GDB, Carson Andorf and Maggie Woodhouse are our primary collaborators. And for the G4 quadruplex work, Hank Bass has been a driving force. So with that, I'm open for questions. Unless you all just want to read my slide.
Thank you very much for your talk. I really enjoyed it. So I know you're working with Mays mainly. When are we going to start having those amazing tools in other species?
So is there a specific species that you're asking about or more generally?
Sorghum.
Sorghum. So this has been something that for many years people have been getting together and saying we need a resource like this for sorghum, right? I would say that you should probably ask the folks that fund sorghum research when they're going to make that happen. I know that there are a lot of people out there that are capable of putting together such resources. So I don't have a good answer for you other than that people are out there that would like to do it. Are there other questions?
What about for soybeans?
So I guess with that, I would direct you to the USDA ARS, that probably talking to Stephen Cannon and David Grant about what their next ideas are would be the right place to go. Are there any other questions?